Welcome back to this next video, and this is the part 2 on the two-dimensional gel electrophoresis. If I give you an overview of the previous video, in the previous video I've told you that 2D page that actually separate the proteins utilizing two of their properties. In the first dimension, the protein, they are separated by their isoelectric point by the technique of isoelectric focusing, and in the second dimension they are separated by their relative molecular weight by the technique of the SDS page. And I told you that it is very unlikely the two molecules they will be similar in two distant properties like the isoelectric point and the molecular weight. So the molecules, they are more effectively separated in 2D electrophoresis than in the 1D electrophoresis. And the advances that has been made in the 2D page, they are actually now separating about 10,000 proteins in a single gel of 20 into 20 centimeter per gel. And there are also advances in the visualization techniques of the protein after they get separated by the 2D gel electrophoresis. I've told you that polyacrylamide gel is used in both of the dimensions and in both of the techniques, like in the isoelectric focusing and in the SDS page. So I've told you that polyacrylamide gel, that is actually made up of acrylamide, the methylene bisacrylamide, which was a cross-linker, the ammonium persulfate or the riboflavin, they were acting as a free radical and they were interacting with the vinyl groups, and thereby allowing the polymerization of the acrylamide and the bisacrylamide, and the timid that was used as a catalyst. Then we discussed about the isoelectric focusing in detail, and I've told you that isoelectric focusing actually separate the protein based on their isoelectric point. So if you are not clear on the isoelectric focusing, you should see my previous video, and I'll share the link in the description. Now in this particular video, I want to focus on the second dimension of the 2D page, which is the SDS page. Now this SDS is actually, this S is sodium, this D is for the dodecyl, and this S is for the sulfate, this P is for the poly, the A for acrylamide, the G is for gel, and the E is for electrophoresis. So it is actually the sodium dodecyl sulfate polyacrylamide gel electrophoresis. For short, it is known as the SDS page. Now the SDS page is a discontinuous electrophoretic system. What I mean by discontinuous is that in the SDS page, the gel is divided into two parts. The first part of the gel, that is the or one part of the gel that is known as the resolving gel, and the other part of the gel that is called is the stacking gel. And both of the running and the stacking gels, they have a different concentration of the polyacrylamide, and this SDS page was discovered by Ulrich. Now the use of the sodium dodecyl sulfate, the SDS, in this particular technique, and the 2-mercaptoethanol, that actually influence the that actually eliminate the influence of structure in charge of proteins, and the proteins, they are solely separated on differences in their molecular weight. So the first thing we will discuss on the SDS page is about this two gel system, about this discontinuous system. If you can see, if this is say for example a single gel, so the first part of the gel, that is known as the stacking gel, which is actually occupying a very small area of the total gel, and then we have got this running or this resolving gel, which is occupying most part of the SDS page gel. Now the purpose of this resolving gel is that in the resolving gel, the protein, they are resolved on the basis on their molecular weight, and the function of the stacking gel is to concentrate the proteins prior to their entry into the resolving gel. So this stacking gel, it has this important function that it is actually staking the proteins on the surface of the resolving gel over here, and when they are stacked over here, all of the proteins, they enter into the resolving gel simultaneously. Therefore all of the proteins, they get equal time for their separation in the resolving gel. Now there are differences between the composition of the stacking and the resolving gel. As you can see over here, in the stacking gel you are using 5% acrylamide, in the resolving gel you are using 12% acrylamide. If you are using a low concentration of the acrylamide, that means that the pores, they will be larger in the stacking gel. If you are using a higher concentration of the acrylamide, the pores that are actually formed, they will be smaller pores. There are also differences in the pH of the staking and the resolving gel. The pH of the staking gel is 6.8 and the pH of the resolving gel, that is 8.8. And this pH of 6.8 in the staking gel, the pH of 8.8 in the resolving gel, that is one of the important things that actually determines the separation of proteins based on their molecular weight. There are also changes in the trace HCl concentration. Here you are using 0.125 molar trace HCl buffer. Here you are using 0.375 molar trace HCl. So the difference between the two, the major differences between the two is that the pH, they are different, and the pores, they are different from each other. Now the differences in the composition of the staking gel, the resolving gel, and the electrophoretic buffer system, as I've told you, this buffer, they are different in the staking and the resolving gel. That is actually making the resolution of the proteins according to their molecular weight. When you talk about the functions of the sodium dodecyl and the 2-mercaptoethanol, this is very important, because if you talk about the proteins, if this is a protein, the native proteins, they can have the positive charge, they can have the negative charge, they may be connected to each other by these disulfide bonds, where the two cysteines, they actually make the disulfide bridges. So this is actually the protein. So if you are separating the protein in this particular format, this means that the shape that will also be playing a role in its separation. But in the SDS page, we are only separating them on their molecular weight. So what we do is that we treat them with the 2-mercaptoethanol. It is also known as the beta-mercaptoethanol, and this 2-mercaptoethanol is actually responsible for reducing the disulfide bonds. And if you can see over here, if you reduce the disulfide bond, that means that the protein, that will be losing its particular folding shape, it will be losing its particular 3D shape. Secondly, when the beta-mercaptoethanol, it linearizes this particular protein, the SDS is going to interact with these proteins, and it is actually giving all of the proteins a negative charge, a net negative charge that is directly proportional to the molecular weight of the protein. So the SDS that actually interacts with the protein based on their molecular weight. If the molecular weight is high, that means a lot of SDS that will be interacting over here. The molecular weight is low, a small number of the SDS that will be interacting with the proteins. So what the the mercaptoethanol and the SDS do, that they are going to linearize the polypeptide, and when they are linearized, they are also having a net negative charge. So the shape and the charge effect of the protein, that is gone, and now the protein, they will get separated from each other purely on their molecular weight. Now this is the structure of the 2-mercaptoethanol. This is the structure of the SDS, the sodium dodecyl sulfate. Now when the protein, they are denatured by the 2-mercaptoethanol, they bind quite a lot of SDS, amounting to their 1.5 gram SDS per gram of the protein, or you can simply say 1 SDS molecule for every 2 amino acids. So if this is a large protein, that means it will be binding a lot of SDS. If it is a small protein, of course it will be binding a small number of the SDS molecules. Now after the SDS, after the interaction of the SDS and the beta-mercaptoethanol, what happens is that the other proteins, they are in their primary structure after the treatment with the beta-mercaptoethanol. So shape does not affect the protein separation. That means that the effect of the shape, that is gone. Another important thing on the protein, that is charge, is all of the proteins, they are negatively charged proportional to their molecular weight after treatment with the SDS, because this SDS is having a negative charge, and when it interacts with the protein, it actually gives a net negative charge to these proteins. So when all the proteins, they are negatively charged proportional to their molecular weight, so the charge doesn't affect the separation. So the shape and the charge effect, that is gone. To talk about the size, the protein gets separated solely on the basis of their molecular weight, that is their size. Now the smaller polypeptide, they move faster because they have to face less hindrance from the resolving gel, and the larger one, they move slower because of greater hindrance. His proteins get separated only on the basis of their mass and their molecular weight and the shape and the charge, that have no effect after the treatment of proteins with the SDS and beta-mercaptoethanol. Another important thing that you use in the SDS page is a bromophenol blue, which is actually mixed with the sampled proteins. That works as a tracking dye in electrophoresis, and that is used to monitor the progress of the protein moving through the gel. So this tracking dye is actually used to monitor the progress of the protein which is moving through the gel. Now when the voltage is applied, the bromophenol blue, it runs along the protein, but it is moving very faster because of its smaller size and because of its negative charge. Now it reaches the end of the gel because any protein, before any protein in the sample reaches it. So when the band of the bromophenol blue that reaches to the end of the gel, that is actually a sign that separation has been achieved, and you have to turn off the electric current. Now even the glycine molecule present in the running buffer, that reaches after the bromophenol blue, and this glycine, that is very important in the separation of the protein and in the stacking of the protein, and it will be cleared in a moment. Now this bromophenol blue, as I've told you, it have got a slightly negative charge, that's why it moves toward the anode, indicating the migration of the protein molecules. Now how actually the proteins, they get separated? So when you talk about the SDS page system, as I've told you, it is a 3-component system. You have got a stacking gel, you have got a resolving gel, which have different pore sizes, and the third important component of the SDS page, that is the electrophoresis buffer. And this electrophoresis buffer, it contains a large amount of the glycine. Now when you talk about this glycine and its stacking function, how it is responsible for stacking the protein in the stacking gel, so the ionization state of the glycine, that is very critical to the separation. Now at neutral pH, when you talk about the glycine at neutral pH, the glycine, that is actually a zwitterion. By that I mean that at neutral pH, it have got a net zero charge, or it is electrically neutral. And at this particular zwitterion state, it have got a negatively charged carboxylic group, and it have got a positively charged amino group. So it have got the negative charge as well as the positive charge. So the net charge on the glycine at the neutral pH, that is 0. Now the pKa of the amino group, that is around 9.6, which is very high than the pH of the chamber buffer in the stacking gel. If you remember in the previous slides, I've showed you that the pH of the stacking gel, that is around 6.8, and here the pKa of the amino group, it is 9.6. Because of this, very little glycine has a negative charge in the chamber buffer or stacking gel, and significant ionization does not occur until the glycine enter the more alkaline pH, which is 8.8 in the running gel. So in the buffer of the stacking gel, the pH is actually 6.8, and in that particular 6.8, it is mostly positively charged, and when it is positively charged, that means it will be very slowly moving towards the positive charge in the electric current. But when it enters the running gel, where the pH is around 8.8, that means at around 8.8 it will be having a very high negative charge as compared to the stacking gel. So it will be very fastly moving in the running gel then. Now this is the say for example the amino acid. It have got this acidic group, it have got this basic group, and in the zwitterion form they have got equal charges, a positive one, a negative one, so it is electrically neutral. Now when you talk about these amino acid and hence the protein, if they are in a low pH from their zwitterion pH, that means at that particular low pH, say for example this is the neutral pH for the glycine, so when it moves toward a low pH, at a low particular pH, it will be positively charged because this amino group is a very good acceptor of the proton, and at low pH there are a lot of protons present. When the glycine it moves to a pH which is higher than its isoelectric point or its zwitterion state. Here it is negatively charged because of this carboxylic group, and a lot of hydroxyl group they are there, and because of that the carboxylic acid group is negatively charged. Now what happens is that when you apply a charge, when the voltage that is applied, the chloride ion in the sample buffer and the staking gel, they move rapidly towards the positive pole, forming the leading edge of the moving ion front. Now you can see over here, if this is the stacking gel, this box like structure over here, if this is the stacking gel, there are three things over here. One, you have got the glycine, and this glycine is actually from the tris glycine electrophoresis buffer which you are using in the stacking gel. This chloride ions, it actually comes from the tris HCl which is actually the running buffer. So when you see over here, the glycine, as I've told you, in the stacking gel, it is mostly positively charged. The chloride ion, they have got the negatively charged. So here actually you are getting a very steep kind of the voltage gradient over here. The positive charge over here, a negative charge over here, but is the space of the staking gel that is very low. So here you are actually getting a very steep voltage gradient over here, and in this particular voltage gradient, the proteins, they are negatively charged, so they are attracted by the positive charge over here, they are attracted by the negative charge, they are repelled by the positive charge of the glycine, they are attracted by the negative charge of the chloride ions. So actually the proteins, they actually stack in the gel. Here the chloride ions, they are, as I've told you, they are negatively charged. The glycine, they are positively charged. Now the proteins that have been made negatively charged, so the chloride ion, they will be having a pulling effect, pushing effect on the proteins. The glycine, they will be having a pulling effect on the proteins. Secondly, you will be having a positive end at the SDS page over here, and when there is a positive end, that means that the protein will also be moving because of its negative charge toward the positive end. So what happens is that is steep gradient of the glycine in the chloride ion that is actually responsible for stacking the proteins into the stacking gel. So once the voltage is applied, the chloride ions in the sample buffer and the staking gel, that move rapidly toward the positive pole, forming the leading edge of the moving ion front. The chloride ion, they will be moving toward the positive end. The proteins, because of the negative charge, they will be moving toward the positive end. The glycine, they are positively charged in the stacking gel, so they will actually be stopping the movement of this protein to a little bit. Hence these proteins, they are stacked in the stacking gel. Now glycine molecule that has a very little charge in the stacking gel, so they migrate at the rear of the moving ion front. Now this difference in the chloride and glycine mobility, that sets up a very steep voltage gradient in the stacking gel that sweeps along the negatively charged protein SDS complex. So this protein SDS complex over here, because of the effect of this chloride ion and this glycine, actually stacks in the stacking gel. Now the large pores of the stacking gel, they present very little resistance to the movement of the protein SDS complexes, which then stack up into a very concentrated region at the interface between the running and the stacking gel. So the protein SDS complexes, they remain concentrated at the interface until the slowly migrating glycine molecule that reaches the boundary of the two gel, because this glycine, because of its mostly positively charged, is actually stopping the movement of these protein SDS complexes. Now what happens is when the protein and the glycine, when they enter into the running gel, a dramatic change occurs because now most of the glycine, the pH is 8.8 in the buffer of the running gel, so they will be now having a very large negative charge over here. Now the negatively charged glycine molecule, they will begin to move at the same rate as the chloride ions, thereby eliminating the voltage difference that controlled protein mobility through the stacking gel. If you can see over here, initially they were positively charged, they were negatively charged, they were negatively charged. So this glycine that was actually having a pulling effect of the SDS complexes toward itself, and the chloride ion, they were actually pulling it downward, and the negative pole of the electric current over here. But when the glycine, they actually enters into the running gel, it again it actually converts into the negative charge, and this pulling effect that is eliminated. Now the glycine, the protein, and the chloride, all of them, they are negatively charged, so they will start moving in the running gel. Now the pores in the running gel, they are much smaller than those of the stacking gel, because I've told you that the acrylamide concentration that is very high in the running buffer, in the running gel, as compared to the stacking gel. So the pores, they present a fractional resistance to the migration of the proteins. Now these proteins, they begin to migrate at different rates because of the sieving properties of the gel. Small protein SDS complexes, they will migrate more quickly than the larger protein SDS complexes. That means they will get separated from each other, and within a certain range determined by the porosity of the gel, migration of the protein in the running gel is inversely proportional to the molecular weight. Greater the molecular weight, the slow they will be moving; the lower the molecular weight, the faster they will be moving. Once you have separated the proteins based on their molecular weight and their isoelectric point, then you are actually going to stain them, and we will discuss about the staining in detail in the coming videos. But this is when you stain the proteins with the Kumasi blue dye, and when you stain them, you can actually see these blue bands which are actually showing you the presence of the proteins in the gel. If you compare the resolving and the stacking gel, so the polyacrylamide concentration in the stacking gel is low, and the concentration of the polyacrylamide in the resolving gel is high, and because of this, there are larger pore size in the stacking gel and a smaller pore size in the resolving gel. The pH of the stacking gel, or the buffer that is used in the stacking gel, that is 6.8, and because of that, the glycine have got a positive charge. The resolving gel that have got a pH of 8.8, thereby having the glycine in the negative charge, and the gradient steep that was made because of the glycine and the chlorine, that is gone, and now the protein, they can actually get separate from each other based on their molecular weight. And the purpose of the stacking gel is only to stake the polypeptide on the interface of the stacking and the resolving gel, so that all of the protein complexes, all of the proteins, they enter into the resolving gel at a single time. And the resolving gel is actually to separate the polypeptide solely on their on the basis of the size. If you like the video, please subscribe to my channel, hit the like button, and share it with your friends.